Well, we'll get started now. Thank you, everybody, for joining us today. My name's Craig Dahlgren. I'm the Executive Director for the Perry Institute for Marine Science. And during this time when a lot of us can't get out on the water, we figured we would try to bring the reef to you through some of these webinars. So we're trying to provide different webinars on a monthly basis. Last month, we did a webinar on stony coral tissue loss disease. And this month, we'll be taking a look at coral spawning. And the webinar will be led by two of our staff at the Coral Innovation Hub at Cape Eleuthera. The Coral Innovation Hub is a partnership between the Perry Institute for Marine Science, the Nature Conservancy, and the Cape Eleuthera Institute. And we're working with other partners at the Reef Futures Lab at University of Miami and Seacorps International as well. Before I turn it over to Dr. Valeria Pizarro of the Nature Conservancy, just wanted to let everybody know with some housekeeping items. We want to keep everybody on mute during the webinar. If you do have questions throughout the webinar, you can ask them through the chat. And at the end, we'll go through those questions and try to address them. Also, we will have this webinar recorded and up on the Perry Institute for Marine Science website later on today or by tomorrow morning at the latest. So if you do have to leave or anything, it will be up there, or if you want to share it with other people if you found it useful, please do so. And with that said, I will turn it over to Valeria from the Nature Conservancy.
Thank you, Craig, for the introduction. We want to thank everyone that is online attending this webinar. As Craig said, I will be presenting, but it will be as well Natalia, that is part with Casey Harris of the Bahamas Coral Innovation Hub. And just to clarify, all the photos that have no credits are from one of the teams, either Team CNC or CI. So during this webinar, we're going to cover these items, focusing on coral reproduction, how we use it for restoration purposes, and especially on coral spawning in the Bahamas. We will have a section of questions, as Craig just said. I'm not going to say that again. And as most of you know, coral reefs are tropical shallow ecosystems, usually found associated to land areas. This ecosystem's importance relies on the ecosystem services they provide not only to us humans, but to a high proportion of marine organisms. Some of the most important are related to fisheries, coastal protection, the pharmaceutical and beauty industries, and the landscape loved by tourists. In the Bahamas, particularly, coral reefs are responsible for the amazing landscape that fuel tourists, providing about 60% of the country's gross product. Coral reefs are built by different organisms, being scleractinian corals or stony corals the main builders. Reef corals have many different shapes and growth strategies, and most of their lives, they are sessile animals, meaning that they live attached to the reef without any possibility of moving. Most of the corals, especially those responsible for building coral reefs, form colonies of genetically identical polyps, where polyps are the coral unit. As a colony, they survive as one big organism, some sharing nutrients through a connecting tissue called cenozoic. Anatomically, these animals are very simple. The body of the polyp might be roughly compared in a structure to a sac, the wall of which is composed by 2 layers of cells, the ectoderm, that is the outer layer, and the endoderm, that is the inner layer. The polyps are really just a stomach, mostly stomach, with a mouth surrounded by tentacles, and these tentacles are used for feeding and defense. Corals grow and build coral reefs by secreting calcium carbonate to form an exoskeleton that holds the colony together and allows them to thrive in turbulent areas. So stony corals reproduce by two means, asexual and sexual. Asexual reproduction includes the budding of polyps, therefore the growth of coral colonies and the creation of reefs, and fragmentation that occurs in some species, especially branching corals, that is an important aspect of their ecology. For different mechanical actions like waves, corals break apart in small pieces that we call fragments, and some of these fragments survive attached to the bottom and grow as independent colonies. It's important to have in mind that during any of these sexual reproduction modes, new polyps or new colonies, they are genetically identical to the mother, like the mother colony or the polyp that buds into two or more polyps. For genetically diverse colonies, corals reproduce sexually. Eggs and sperm are produced, fertilization and the development of larvae occurs. This is the only time in the coral life cycle they are free-swimming organisms. The larvae, when completely developed, search for a place to settle. When the cues are found, the larva will settle, become a spot, and metamorphose and produce a rudimentary exoskeleton and becomes a recruit. From there, the first polyp grows, divides, or buds into more polyps, and with time, it becomes a juvenile and a colony, and then the cycle starts all over again. As I told you, the anatomy of the coral is very simple. However, coral ecology and biology is more complex. For example, corals have two different reproduction modes, brooders and broadcast spawners. Brooders, that are the ones that are just highlighted in red, include all coral species that have internal fertilization. In this case, the sperm is released to the water column and currents and waves transported to other colonies. The colonies capture the sperm and fertilize the eggs that they have. Larval development occurs within the colony and it is released to the water column almost ready to look for a place to settle and recruit. So it only takes few hours or few days for the larvae to start looking for a place where to settle, become spot, recruit juvenile, and then the cycles begin again. Broadcast spawners release eggs and sperm to the water column where fertilization occurs. Some coral species are hermaphrodites, meaning that in one colony they can produce both eggs and sperm. And other species have separate sex colonies. Additionally, in the case of the hermaphrodite corals, some species produce both female and male gametes, that is eggs and sperm, at the same time, and they put them in a package that we call gamete bundles, while other species produce 1 year eggs and the following year the sperm. All broadcast spawners release gametes into the water column where fertilization and larval development occurs. It takes, in this case, not few hours, but usually it's more like few days, 2 weeks to actually the larvae to develop and to start looking for a place, become a spot, a juvenile, and then the coral. To increase fertilization rates, corals from the same species, especially in the Caribbean, usually spawn the same day and time, as Natalia will explain later on. To understand why we study coral reproduction, coral sexual reproduction for restoration, we need to know about the current status of coral reef ecosystems. And the Caribbean coral reefs are home to approximately 44 million people, most of them living in coral areas and depending on coral reef ecosystem services. Unfortunately, coral reefs are deteriorating. This map is from the book Reefs at Risk, and it shows which reefs have higher risks of disappearing in the following years. In dark red and red are the ones with higher risk. And as you can see, Caribbean reefs are particularly at risk. It has been estimated that since 1950, we have lost about 50% of coral reefs in the area. Bahamian reefs are suffering the same fate, as you can see in this figure from the report card for the Bahamas. In red is the percentage of reefs in poor condition, and in blue, the ones that are in good condition. The majority of the reefs assessed in some of the islands of the Bahamas are in an impaired condition. This means that those reefs are not functioning properly, thus not providing the ecosystem services. So knowing the importance of these ecosystems, we are using ecological restoration principles to recover Bahamian reefs to a point that they can function and provide us ecosystem services that we rely on. And as Craig said, during the last 2 to 3 years, the Nature Conservancy, Peru Institute for Marine Science, and the Cape Peru Institute have been implementing 3 strategies to restore or recover Bahamian coral reefs. Our approaches are larval propagation, that I will explain in detail next, growing coral fragments in coral nurseries, something that the Peru Institute for Marine Science, along with the Reef Rescue Network, have been doing with other partners in many islands throughout the Bahamas, and microfragmentation that the Peru Institute for Marine Science has implemented as well in New Providence. So as I said, I will focus on how we use sexual coral reproduction for our restoration purposes. I am showing you this slide again so you can remember or be reminded of the different phases of coral reproduction. So again, we need to know exactly when the corals are spawning, and we need to know as well how long the development of the larvae takes, so they can start looking for a cycle, and then how long it takes to become a spad or a settle, and how long it will take to become a recruit. This allow us to actually go to the lab, as I will show you, and work with all the different species. We are working right now, are focusing on broadcast spawner species, because these are the main coral reef builders, and we want to restore or recover reefs. We need to work with these ones that are the main ones. So when we know when corals reproduce, we can go actually to the reef and collect their gametes. So what we do is we place these things that we call gamete collectors. We place them on top of the corals that we know they are going to reproduce. And Natalia will explain you exactly how you know when they're going to spawn or release the gametes. So in this case, and most of the cases, the corals that we work on, they release these gamete bundles. These gamete bundles are buoyant, so they will start going to the surface, and we have a vial on top of the collectors. So when we have enough gamete bundles on these vials, we just transport them into the lab. In there, what we do is we separate the sperm from the eggs of the same colony, and we do cross-fertilization. That means just taking the sperm from one colony and mixing with the eggs from another colony. This allows us to do what? This allows us to first avoid self-fertilization, that is when you're mixing the sperm and eggs from the same colony. And we try to avoid that because we don't have, like usually the embryos and the larvae and even the recruits that are from surface fertilization have very low survival rates. And the diversity, especially the genetic diversity, is very low in those cases. So what we want to do is we want to do cross-fertilization so we can increment or increase the genetic diversity of the corals that we obtain. So we have them, usually the larvae or even the embryos, until we are in the lab. However, before we do that, and especially in collaboration with other organizations like STAGHORN International, few weeks before spawning, we have to leave the substrates in the water. And these substrates that are in this picture here are the ones that we provide the larvae to settle. Remember, in the cycle, the larvae starts to look for a place where to settle and recruit. So if we want to do any restoration, we have to provide those substrates. So we leave those substrates for at least 4 weeks so it can create a biofilm growing on them. And that biofilm will have the chemical cues for the larvae to actually go, settle, metamorphose, and produce the first polyp. The other thing that we do, and that is actually a few days before spawning, is set up what we call, and is these blue things over here, what we call a floating rearing pool. That is a structure where we can place the substrates, as you can see in this picture, and then the coral larvae in the sea, or in this case, in the marina of Cape Eleuthera, the Cape Eleuthera marina. With this pool, we aim to upscale the restoration process. When the embryos are fertilized or the planula is fully developed, we take them to the pool. As you can see here, we just put them there. This pool doesn't allow anything to go in or anything to go out, like the larvae, but it allows the water movement, so it's better for the larvae to be there. We check them daily, and then the first days you will see them just all swimming around, and after a few days and you start seeing that the number of larvae swimming around decreases, you go and take some of the substrates to the lab and look under the microscope, and you will look for this. There are the spots of the corals, and if you're very lucky, after a few days, you can have like recruits like in this photo where they already have the cells and telly. When most of the corals have recruits, what we do is we take the substrates to the reef to outplant them. And what we want in the following years is to start seeing and watching the corals just growing out of the larval propagation strategy. With this restoration strategy, we aim to upscale coral restoration and to increase genetic diversity of the coral populations to make them more resilient in this changing world. And I'm going to stop here. I'm going to stop sharing my screen. And Natalia, you can share your screen now. Thank you.
Hello, everyone. Valeria, could you please confirm if you can see my screen?
Yep, I can see it.
Perfect. So, well, thank you so much for joining us today. So Valeria already explained sexual reproduction in corals. So now we will focus in the moment when the corals release their eggs and sperm, called gametes, into the water column. One sec. Oh, okay. So in this picture, you can see those pink dots, which are eggs and sperm packets or bundles that are released through the polyps. And basically, the first thing that we will see is that the coral gets ready to spawn. That's called setting. So basically, pink or orange dots appear in each polyp, and after a few seconds or minutes, depending on the species, the coral will release them. So here we have in the red square, this is amazing pictures taken by Ellen Mueller in Bonaire. So in the red square, we have two species that release gamete bundles. So as you see, corals are different, and so their gametes. They can have different sizes, different colors. And on the right, we have a gamete bundle from a brain coral. This is a close-up where you can actually see the pink dots and some white stuff, which is the sperm. So that's the gamete bundle. Some corals release eggs and sperms separately. So here in these pictures, we have a male colony releasing a sperm and a female colony releasing some eggs. But how can we know when? So coral spawning is synchronized with the full moon, with the sunset. They have a specific time of the year. Depending on the coral species, they also have specific times. It could be at night. It also could be during the day. So scientists and divers have been documenting these spawning events for decades. coral spawning is really precise, and now we know that some species in some areas release their gamete bundles at the same time. But we see a lot of observation in the Bahamas, so we don't have enough observations, so that's why we're doing this. We really need this information because we want to improve our restoration efforts, and the best way to do that is having a lot of ice in the water. So if you can dive during the predicted times and dates, it will be awesome. So as I said, corals synchronize with the full moon. So for example, this year and this month, full moon was the 7th of May, but last year was the 18th. So that's why we don't rely on dates. We just focus on the full moon. So for example, if you observe a coral spawning 5 days after the full moon and 10 minutes after the sunset in September, it's highly possible that you will observe the same the next year. So that's how we have these observations right now. So corals, as you see here, so corals are different. They grow at different depths. They have different shapes. And at the same time, they play different ecological roles on the reef. So we have fast-growing species, like the branching one in the middle. We have slow-growing ones, boulder and massive corals on the right. And I'm talking about this because that also means that they have different reproduction and life strategies. So for example, here we have some common species in the Bahamas. We also have the months when they spawn. We also have the numbers. The numbers that we have, those are the days after the full moon when they're spawned. And those numbers in red, that's the peaks for each species. So as you see, the first coral on the left, the group brain coral, that one grows few centimeters per 1 or 2, but that species can spawn in 6 times or maybe more. But on the right, we have the famous STAGHORN and ELKHORN coral. These corals can grow something between 10 and 20 centimeters per year, but they spawn just once. So you can actually see that they invest their energy doing different things. So I'm going to start now with this beauty. This is a group brain coral. This one is one of my favorite ones. It's really easy to identify. It's really abundant. This one spawns several times per year. It's also during the day, which is great because you don't need to be an advanced diver if you want to observe the spawn. I'm going to show you. Well, I hope that you can see my screen. So the setting time for this species is really short. It's also before the sunset, so you can just go to the field during the day. In some areas, you can observe some butterfly fish that can alert us when or which colony is going to spawn. This video was made by Reef Renewal Foundation Bonaire last month, and you can see how the fish is eating the gamete bundles. So those bundles have lipids or fat, which make them buoyant, and I guess also make them really tasty for the fish. And so as soon as the coral release them, they will float and they will go to the surface, as you can see. Okay, now here I have 2 different brain corals. So they spawn in August, September, and October, as far as we know. The peak is the 6 and 7 days after the full moon. And it seems that we have 2 different groups, one early and one late. But both groups release the spawn after the sunset. That means that you need to do a night dive if you want to observe these ones. So it could be a lot of worms in the water, so you actually can see the coral eating them, which is really cool. The setting time is really short. For example, here I have the great star coral. This one is also really easy to identify. It's really abundant. This species release eggs and sperms separately. Actually, here I have a male colony releasing a sperm. It looks like it's smoking, at least that's how it looks to me. So something important, not just with this species, with corals in general, but they expose their tentacles at night. So maybe they can look a little bit different. So you just need to be aware of that they just look different. And you can also see them eating worms during the night dives. So the peak is also 6 and 7 days after the full moon. And it seems that we also have an early and a late group, but also after the sunset. So here we have the lobed star coral. This one released gametes in September and October. It could be that they also release in August, but we don't know. The peak is also a 6 and 7 day after the full moon. The setting is a little bit longer than the brain corals. Actually, here you can see a picture of the setting. So you see, like, it looks like orange dots. Almost the whole colony is ready to spawn. For this one, you also need to do a night dive, but this one is quite late. It's like after 9:30 p.m., more or less. Here we have the mountainous star coral. This one spawned the same months as the previous one. That means September and October. Also, a peak is 6 and 7 days after the full moon. Also, you can see the setting. It could last some minutes. I'm just going to show you another video. Okay, so here we can see the setting. So the dots or the bundles looks like more orange when they're like still in the mouth of the polyp. But when the colony releases them, they look like more pink. They also have longer setting times than the brain corals. This video was made in Colombia by ELKHORN in a collaboration with Valeria. So if you go to a place with high abundance of these species, that will be really amazing. It looks like a ocean as a snowstorm, which is really, really cool. Okay, and now the last two species, but not least, we have the ELKHORN and the STAGHORN coral. So they're really common to find them in shallow areas where the wave energy feels way different than in deeper zones. So ELKHORN coral is found in August, the 3rd day after the full moon. But what is different with this species is the setting can last like around 20 minutes. So for example, if you observe a brain coral, that's just a matter of seconds, where you see the gamete bundles, like when you see the setting. But with this one, you can wait 5, 10, 15 minutes. So don't worry and just wait for it. This one is an endangered species, and diving around them requires experienced divers because we don't want to break them. Actually, we're working really hard to restore them, so we don't want to destroy the corals. So the same goes for the STAGHORN coral. This is also an endangered species. Their spawning predictions are really unclear. So for this one, we have some observations the 7 and the 8 day after the full moon in August. But in other places in the Caribbean, they saw the spawning even the same day with the full moon, first day, second day, 10 days. So having observation of this species will help us a lot to improve our restoration efforts. So with this one, we're not completely sure. And now, how can we know where and when? So here, so the first thing is to define the location. So here as an example, I have Eleuthera, that's where we are located. That's in the red square. So you can use the website, which is at the bottom, to find the moon phases and the sunset times. So depending on the species, for example, if you want to have a look at the group brain coral, and I just told you that that spawns something between 10 and 13 days after the full moon. So you have a look, okay, when was the full moon? So that was, oh, this is in Spanish, sorry about that. But this is the 7th of May, and then you have to count 10 days, and then you just need to have a look, okay, what is the sunset time for that day? And then you can make your calculation. So here we have the spawning predictions for South Eleuthera. So we have in red the spawning peaks. We have the sunset for each day. We have the dates, the spawning windows. We have the species, common names, scientific names. But you don't really use this information for whole the Bahamas. And why not? Because the Bahamas is quite big, and we have half an hour difference between the sunsets. So for example, here we wrote down an example of the sunset of today. So we have, I just choose 3 random ones. So we have Bimini, Cat Island, and Little Inagua. So the sunset time is different. So what we did is that we divide the Bahamas in 3 different zones. So we have zone 1, zone 2, and zone 3. Just for making things easier, we kept 10 minutes different between them. So don't worry, you don't have to make any calculation, you don't have to count. We already did all of that. So we have here the suggested monitoring dives. We have the dates, the months, we have the dates, we have the targeted species for each date, how many dives we recommend to do, the dive time, and here you can see, you can just, depending on your location, you see if you're located in zone 1, 2, and 3, and then you just start your dive in the suggested time. So as you remember, a lot of species spawn like during the 6th and the 7th day after the full moon, which is the 7th and the 8th of September in this case. So those days are ideal to do like 2 or 3 dives because the species, they spawn at different times. So here we just make like some groups so you can just have a look of different species during those times. So it's not the same case with the group brain coral, which you just need to do 1 dive. It could be a little bit longer if you want. So here as our recommendations, so during the night dives, so the first thing is always bring a dive light with you. Even if you're doing a monitoring dive for the group brain coral, which is during the day, just bring a dive light just in case it gets a little bit dark, just for safety. Also, it's good if you bring an extra light. If you have compass, full wetsuit, camera, glow sticks, slate and pencil. So why do you need a slate and pencil? Because that will be super useful for us. If you can take some notes about the setting times, spawning times, monitoring dives. As a recommendation, the best thing to do is if you can dive in a familiar dive site so that you know the area really well, and also just make sure that the coral species that you're looking for is in that area and at that depth. Also secure all your materials, so I don't want you to lose your camera or dive light because you're writing something. You can also use glow sticks as a references, for example, for the descent, for the ascent, for the exit, for the entrance. You can set dive computer and the camera with the local time. So as you notice, there's some time differences. So important, if you don't do night dive too often, just make sure that you practice the signals, that you know how to use your dive light, your gauge, compass. Also, you can make your own signals, for example, for setting and for spawning. So here as an example, you can just put your hand with your fingers facing down for setting and facing up for spawning. This is just an example. You can just create your own, that's really up to you. During the night dives, you just need to be careful with things that you can see. For example, jellyfish, also maybe some sea urchins, they're really active. Keep an eye on your dive body the whole time. Check your time, check your air consumption. Super important, safety is first. So plan your dives, respect the dive plan, respect the depth, bottom times. Also, if you don't feel comfortable or if it's something, just cancel the dive. So remember, safety is first. Be careful with your buoyancy, especially in the shallow areas. Be careful with your fins. Also check your dive gear first, and aid kit and oxygen if you have them. Stay safe and of course have fun. So monitoring dives, they're super nice. There's a lot of things to see. You can imagine like how many things are going on. So first thing, adjust your personal watch with the local time. Also, it's good if you can find a shallow area with high coral cover. So for example, if your dive will be 1 hour, it's just better to cover in a small area. Just keep an eye on the target species that you're looking for, and you can just do the whole dive just making circles, just keeping an eye on the same area. So, but there's also other reef creatures spawning, for example, soft corals, brittle stars, Christmas tree worms, fireworms, sea urchins, sea cucumbers. So there's like a lot of activity on the reef during those days, especially in September and October, around the 6th day after the full moon. So here I just put some really nice pictures of Ellen Mueller. I highly recommend you to check her website. So this is just for give you like an example of the kind of things that you can see. And of course, that depending on the size of the species that you're looking for example, with the brittle stars, the eggs, they're really tiny. Or the same goes for the Christmas tree worms. These are really tiny eggs. So keep your eyes open because you can see a really nice stuff like these brittle stars. It seems that they're having a lot of fun. So for the spawning observations, so here's a form that you can fill if you decide to do monitoring dives. You can send us a picture of the data Or for example, if you dive during the suggested times and dates and you don't see anything, please also let us know. That will be really important for us to make our predictions for next year. If you're not sure about the species, also you can just take a picture. So here, don't worry if you don't remember or if you didn't count the full moon and that stuff. Just write down the date, location, dive site, depth is also important, area, and for example, just write down the name of the species. It could be a common name, and then you can just, if you have bundles, egg, sperm, also how many colonies you saw spawning, how many colonies you monitor. That's what the importance of this is, because if you decide to go to an area and it's not so many of that species that you're looking for, so it's important for us to know if you saw just one colony during the whole dive or if you saw 20. So that's why it's just important to keep an eye on those numbers. It could be like an estimation. Important also to write down the setting time, spawning time, and also at what time stop. Other thing that I forgot, if you see one coral just spawning, just keep your eyes open, because it's highly possible that corals around will start doing the same. If you have any questions, spawning observations, if you're not sure, if you need help planning your dive, just send a message to us. So here we have our emails. So you can just send us pictures or information or questions. So, well, thank you so much. And so Casey, Valeria, and I, we are really proud mamas. So that's why we like to show our babies. So here we have some Poraites recruits and some of the Orbicella violata or the mountainous star coral.